नमस्कार दोस्तों, welcome to channel Toxicology on board. In this video, we will be learning about the topic DABT topic clastogenicity. Let's directly dive into the question. So the question is, what in vivo assay is designed and used primarily for the detection of chromosomal damage clastogenicity? Okay. So this is a typical micronucleus slide. Okay, here these are nothing but micronucleated polychromatic erythrocytes, and these are normochromatic erythrocytes. Okay, so let's see what options are there. First, the question we need to again understand. We have to identify what in vivo assay is designed and used primarily for detection of chromosomal breakage or clastogenicity. So what are the assay for clastogenicity we need to identify. Okay, we have option A Drosophila sex-linked recessive lethal assay. Option B we have rodent bone micronucleus assay. Option C TK assay in mouse lymphoma cells. And option D chromosomal aberration assay in human lymphocytes. The right answer is option B, which is rodent bone marrow micronucleus. So what is the explanation and the difference which is given by ABT? The rodent micronucleus test is the only in vivo assay for detecting clastogenicity. You have to understand when it comes to identification of mutagenicity, genotoxicity, clastogenicity, all these terms I will explain in another video, all the definitions also. Okay, so there are different battery of genotoxic tests will be there. Okay, in that main important thing one has to remember or understand in detail how the even the principle and the method how the assay is that AIMS assay first you need to know salmonella typhimurium assay, then chromosomal aberration assay one has to know, and also micronucleus assay. So this video is about micronucleus assay. So micronucleus assay is specifically to identify clastogenicity type of DNA damage, chromosomal damage you can say. So two chromosomal aberration assays, one in a Chinese hamster ovary cells and other in a human peripheral blood lymphocytes. Okay. Um, are also used, but they are in vitro assay. So this particular rodent bone marrow micronucleus is the only in vivo assay. If the same question comes for in vitro assay, then you have to remember this option CHO, that is Chinese hamster ovary cells, as well as human peripheral blood lymphocytes. Okay. Then next, the mammalian microsome reverse mutation assay is like the AIMS assay is intended to detect mutagenicity. So you have to always remember AIMS assay is to identify mutagenic damage, and this micronucleus assay is to identify clastogenicity. Example: point mutations, forward mutations. Again, in mutations also different types of mutations. Frame shift mutations will be there. In that again deletion, addition. Such type of damages will be there. So it is used for point mutations and forward mutations also. What your aims is it? Okay. Then let's see. So these were the questions or explanation which was given by American Board of Toxicology. Then let's see what else we can learn from this concept. So the rodent marrow micronucleus is a standard in vivo test. Okay. Other two options were CHO cells as well as other option was about in vitro test used to detect chemical that causes chromosomal damage, that is clastogens, or damage to the mitotic spindle, that is aneuploidies. Okay, it works by this micronucleus assay works by examining micronuclei, which are small extranuclear bodies containing chromosome fragments or whole chromosomes in newly formed red blood cells, reticulocytes from the bone marrow of from the rodents. So this is the smallest one. See this small with arrow you can see, this one is your micronucleus. This assay is a crucial part of genotoxicity testing. So initially when during the drug discovery screening also, AIMS assay is as usual, it is the main important. If AIMS positive is there, next what two other essays we have to do is like that. This essay also comes one in that case. So this essay is a crucial part of genotoxic testing for pharmaceuticals as well as chemicals, helping to assess safety and potential health risk. So if you look at this is a typical diagram, you can see that whatever the test item is given into the test system, here it is mice, and you will isolate the femurs. From this particular, after once the animals are sacrificed, you will we will take out the femurs, and these femurs are flushed in this bovine serum, I guess, fetal bovine serum, I guess. Then this you will be mixing this bone marrow cells with FBS. Then you will be preparing slide preparation will be there, smear preparation will be there. Then the fixation of the slide with the methanol will be there. Then we will be subjecting this particular slide to GIMSA staining. Then we will be analyzing after this smear is fixed. Then you will be analyzing. You will be reading under microscope. So let's see in another slide what else we can learn from this particular concept. So how this micronucleus assay works is exposure to the test agent. Here the test agent may be either a chemical or a pharmaceutical. Rodents, mice or rats are exposed to the chemical being tested, as you can see in the previous picture. Bone marrow collection. Then you will collect the femur from the rodents at specific times after exposure. Then reticulocyte identification. You have to identify that RBC. Bone marrow contains immature newly formed red blood cells called reticulocytes. Micronucleus scoring. Reticulocytes are examined for micronuclei, which indicate that a chromosome or chromosome fragment was left behind during cell division. During the mitotic division, no, the small fragment will remain in this newly formed erythrocyte. This newly formed erythrocytes are easily to identify because they stain positive for the RNA content. Okay. So generally, if you see enucleated should be RBC should be enucleated. Okay, complete nucleus will be used out of that particular newly formed cell, but the small particle, no, it will remain. So this is your micronuclei. The frequency of micronuclei in the reticulocyte is counted. Okay, the huge number, like I think around thousands of counting of these cells will be there. In that you will be identify ratios. You will PEC by PNEC, like this ratios we will be doing. Um, so the frequency of micronuclei in the reticulocyte is counted and compared to control groups to determine if the test agent caused genotoxic damage. So here in the picture you can see this is an erythroblast, and here this particular RBC cell is subjected to genotoxic damage. As a result, what happened? This nucleus is split down into two. One here you can see clearly the formation of micronucleus polychromatic erythrocytes. Okay, nucleus is expelled out, but that small micronucleus still remain in the newly formed the polychromatic erythrocytes. So when you're reading your slide in the microscope, you need to identify these type of cells. Okay, so the one which is not at all damaged with genotoxic damage, here you can see the nucleus is intact and the complete nucleus is expelled out during this mitosis, during this nucleus expellation. So this polychromatic erythrocyte, it contains no nuclei. So this is enucleated erythrocyte, you can see. So these are the various stages you can see. Okay. So, what are the key characters of a micronucleus assay we will be seeing? One is target cells. Primarily, this micronucleus assay uses immature red blood cells, newly formed reticulocytes from the bone marrow. Endpoints we are able to detect by using this micronucleus: clastogenicity, that is chromosomal damage, and aneuploidy, numerical chromosomal changes. And coming to applications of this micronucleus assay, it is widely used for assessing genotoxic potential of new chemicals, especially in the pharmaceutical industry. As I said, no, it is a part of battery of toxicology genotoxic test. On its aims assay, chromosomal micronucleus is also very important because it is used to identify clastogenicity. So why it is important? Because it is a regulatory requirement. It is requirement by either by the FDA or EMA. So this is a we need to perform for any new chemical. We need to have this particular data. So it is a regulatory standard. Then it is used for hazard identification also. So whenever there is whenever you are doing performing any risk assessment, there will be certain endpoints will be there: genotoxic hazard, carcinogenic hazard, sensitization hazard. So you will be looking, you will be fetching for the data of that chemical with respect to this particular assay, whether it is positive or negative. So for hazard identification is also very very important, this micronucleus assay. Then we have mechanism of action. We will be able to identify the mechanism how this particular chemical is causing the chromosomal damage. Okay, and ease of scoring. I won't say it is easy of scoring. Based on my experience, I say it is laborious task. You need to read lot of slides, and you have to you have to count lot of cells, a huge number of cells you need to count throughout that particular slide. Okay, let's see. Let's understand better with the example of another picture. Here is the cell, and it is causing DNA damage. So after subjected to cytoskeletons B, you can see the split of nucleus has taken place, control binucleated cell without micronucleus. And here, after subjected to mitosis, you can see two cells. Cell is splitted with two different nuclei, but one of them is also containing micronucleus. So micronuclei induced by genotoxic damage. This nuclei is induced because it is causing damage to this nucleus, because of which a micronucleus is formed. So we will be counting these cells, okay, while when you are doing scoring of the micronucleus slides. Okay. Hope from this video you were able to understand the importance of this micronucleus assay, how it is used to identify clastogenicity. You need to remember, as I said, no, you have to learn about battery of genotoxic test. You should be learning. You should at least 3 essays, because when I look into Clayson textbook, no, more than I think 40 plus essays are available with respect to genotoxic assay, and all these are also listed. Many of them are also listed under OECD guidelines. We have series of 470 say started from that you have 472, 473. All these things essays are also related to genotoxic damage only. I believe those who are working in genotoxic area, they will be having in-depth knowledge about each and every essay. But other general toxicologists might not be working with these essays on a day-to-day basis. So at least they should be knowing these 3 essays. Okay. Also remember the guideline number. Which OECD guideline number is for which type of essay. So hopefully in another video I will list out all the OECD battery of genotoxic guidelines and their number across this that OECD number like 470, which guideline name, that I will list out. So it will be very easy for you people, or you can go to OECD website and you can download there will be a PDF where you will be having test guidelines, list of test guidelines. In these 4 sections will be there. In this health will also be there, health effects will be there, physicochemical characteristics will be there, ecotoxicology will be there. So in that you have to read about health. Okay. Being a toxicologist, you should also be aware of ecotoxicology guidelines are also there in the OECD test guidelines. I think that is 201 series will be there. So, so thank you so much. Be curious. For any questions, please contact to my mail ID arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel Toxicology on Board. Thank you so much. All the best for your exam.